I'm Daniel Luksic, a medical science liaison with Natera's Organ Health Division. Today I'm going to be presenting on a paper titled The Phenotypic Difference of IgA Nephropathy and its Race and Gender-Dependent Molecular Mechanisms. This paper was published in Kidney 360 in 2021 by a doctor named Suzuki. IgA nephropathy is caused by deposits of immunoglobulin A antibodies in the glomerulus of the kidney, where it leads to inflammation, scarring, and eventually hematuria and proteinuria. This is a very heterogeneous condition and can be thought of as a symptom of several different types of disease rather than a disease of itself. It's also very common, with up to 10% of healthy autopsies and up to 17% of healthy donors having IgA deposits found in their kidney. This paper focuses on the heterogeneity of IgA nephropathy in different populations, specifically East Asians and Western Europeans. IgA nephropathy is much more common in East Asia, partially because they have population screening for kidney disease as a much more routine part of practice there. The demographics of it is also markedly different in each region, with East Asia having an approximately 1-to-1 male to female ratio, whereas in Europe and the United States, males are around 6 times more likely to develop this condition. Now, the mechanisms that we know about are that the galactose-deficient IgA1 forms complexes and precipitates in the kidney. These complexes can be found in the serum, and they can actually predict outcomes, but East Asians who develop IgA nephropathy have very different levels than seen in Europeans. Furthermore, we do know that there are certain genetic correlations in both populations, but novel mechanisms that are only seen in East Asians suggest that there's a different mechanism for these IgA deposits forming. This study was designed as a chart review of patients from hospitals in Japan and Europe in order to compare the outcomes and the clinical features that was found in IgA nephropathy patients in each region. In total, there was 907 patients which were reviewed, approximately 50% from Japan and Europe. And although there were many differences, there was also a lot of similarities here. Some of the most marked differences comes from the fact that the Japanese patients were more likely to have been discovered with their IgA nephropathy from routine clinical care. As we can see in the bottom here, the highlighted section shows that 74% of these Japanese patients were discovered as part of a systematic screening, where only 10% of the European patients were. This corresponds to some other trends in the data, such as the eGFR of the European patients generally being lower, and there being a quicker time for the European patients to get biopsies because they were more or less presenting when they were more advanced and they were more sick. In terms of the presentation of the disease itself, they were pretty similar between the two different cohorts, with the Japanese and European patients having approximately the same amount of persistent macrohematuria between them. The most stark difference between these cohorts is seen in the gastrointestinal comorbidities. Overall, 17% of the European patients, but only 1% of the Japanese patients, had some gastrointestinal comorbidity such as celiac, Crohn's, or ulcerative colitis. Furthermore, though we did see elevated IgA levels in the serum of all patients with IgA nephropathy, the Japanese patients much more frequently had highly elevated IgA levels in the serum, but had much lower GI complications. Additionally, in terms of the treatment is another place where we saw marked differences between the two cohorts, with the Japanese patients much more likely to have received tonsillectomies as part of a course of treatment of the IgA nephropathy. They were also much more likely to receive corticosteroids. IgA nephropathy coincides with upper respiratory tract infections in both patient populations. However, as we mentioned before, the gastrointestinal comorbidities are very different. This may be due in part to the mechanism that IgA uses to interact with the gut. There is a molecule called polymeric IgA receptor, which ferries the IgA molecules across into the intestinal mucosa. And mouse models have given us a clue to where this system can become overwhelmed, leading the IgA to build up in the serum and eventually depositing in the kidneys, causing IgA nephropathy. There have been many studies of European patients showing that sensitivity to gluten, even at a subclinical level, is strongly associated with IgA nephropathy. And in fact, mouse models have been able to clearly demonstrate how administering or restricting gluten from the diet will dial amounts of serum IgA levels up or down. However, it's very rare for IgA antibodies to be targeted against gluten and other allometric antigens, whereas in the European population, this seems to be common. And this source of discrepancy between whether or not IgA is sensitive to gluten and other dietary compounds is something that may result in the vast differences that we see in the IgA nephropathy between the two populations. The PIGR transport molecule may also be involved in an explanation for why the European cohort has such a marked difference between the sexes in the prevalence of IgA nephropathy. Estradiol actually is found to augment the PIGR activity, which may give rise to the differences that we see in men and women. There have also been certain SNPs that were identified, that single nucleotide polymorphisms, genetic markers that correspond to a risk of IgA nephropathy, but also response to the tonsillectomy as a treatment, as well as a decreased risk of certain gastrointestinal disorders. However, in certain European populations, this SNP has actually been shown to be associated with increased risk of certain gastrointestinal disorders. Some other mechanisms that have been elucidated in genome-wide association studies have been involving ligands called APRIL, which is involved in the IgA accumulating in the kidney. And new research is being done that elucidates the molecular mechanisms of the immune response mediated by the tonsils as it relates to IgA levels. In conclusion, the differences in IgA nephropathy between the two different regions of the world suggests that there is a combination of environmental and genetic factors that may play a role in the different prevalences, the different comorbidities that we see in this condition. However, there's still much to learn about this, and more research is needed before we can really truly have an accurate understanding of the mechanisms of IgA nephropathy in different patients.